Hello and welcome everyone. Thank you so much for registering for the webinar on Unravelling the Transcriptome using RNA sequencing. Our speaker for the day, Dr. Ambily Sivadas, has already joined us. She is a welcome DBT Early Career Fellow working at the Division of Nutrition SGRI in Professor Kurpat's lab. She is a computational biologist by training. She obtained her PhD at CSIR IGIB with Dr. Vinod Skaria, where she developed novel approaches to advance pharmacogenomics using population-scale genomics. She has extensive experience in integrating different omics approaches such as genomics, transcriptomics, metabolomics, and epigenomics to test different biological hypotheses. Her current research interest spans pharmacokinetic modeling, pharmacometabolomics, and population genomics. We welcome you, Dr. Ambily, and over to you for the webinar. Thank you all for joining in.
Are you able to see my screen? All right. Okay, so good afternoon, everyone. I mean, really happy to see that so many of you have come to attend this webinar. And as I said before, anywhere you have a query or something that would need a little more elaboration, please feel free to interrupt me and ask me questions. So in today, we are primarily going to give a quick overview of what transcriptomics is all about and what's the real rationale behind doing RNA sequencing, and also to understand a little bit about the experimental workflow, the basic experimental workflow that is involved in an RNA sequencing experiment, as well as also in the data analysis side. And finally, I'll also be sharing some of the useful RNA-seq resources that will be good to expand your interest in this area. So without much delay, let me just jump in. So first of all, what is it that that motivates all of us to learn beyond DNA into other different levels of regulation? So we are all aware of the central dogma of biology, which is nothing but DNA replicates and it transcribes information into RNA, which then translates into protein, and which are the machinery that runs the different functions of the body. But now, with decades of research by the scientific community, we now know that it's not a simple chain of events as described in this one. We have an extended central dogma of biology now, with different regulatory players involved. So I'll just briefly try to take you through these the different levels here. So let's start off with a gene that's on the DNA, and we all are aware of the exons, the coding exons, and the introns in the gene, and then the regulatory sequence that's upstream of a gene, which plays a role in governing the extent to which a gene is expressed or not. So there are there's the RNA polymerase, which binds to this upstream regulatory sequence, which also again manifests its function by interacting with other proteins like the transcription factors and regulatory proteins. So at the DNA level itself, one can easily see how different regulatory players or molecules are involved in deciding how or to what extent or which genes transcribe. Now, once the gene gets transcribed into the primary transcript, then it undergoes a differential splicing process, which we again all of us know, where the different exons are stitched together to create different isoforms of a gene, which are known as transcripts. And these different isoforms can be expressed to different levels. And now the mature RNAs then later go on to the translation process in the presence of ribosome and other regulatory proteins, and we have the proteins. Now we also know that the modifications or the regulation doesn't end there. Even after translation, there are post-translational modifications in terms of phosphorylation, acetylation, prenylation, or different chemical modifications that happen at the protein level, which can further alter or modify the function of the proteins that are created from the gene. All right. On the other side, again, as far as DNA is concerned, there are other levers of change that can be brought in terms of, again, chemical modifications like methylation on the on the nucleotide bases, which are referred to as a DNA methylation, which can again decide on which genes and to what extent the genes can get expressed. And we all know that this also manifests in the form that chromosome or the chromatin is packed and which genes are made accessible. So then the chromatin patterns which are bounded around histone proteins, again these proteins based on the chemical modifications, the ones that I discussed before, can again have an impact on the accessibility of a gene for the transcriptional machinery, thereby again having another level of control over the expression of the genes. So I think this in this few minutes we know now the different levels of regulation or the different levers that can control the gene expression of a gene, and the which gene as well as to what extent each gene can get expressed. Now, understanding each level of these regulation requires the use of different kind of techniques. There are epigenomics, which involves everything involved with the DNA methylation, then the ChIP-seq, and to look at the histone modifications. You have transcriptomics to study the RNA, the set of all RNAs that are there in the cell. Then you have proteomics to look at what happens at the protein level. So there are a wide range of techniques that we use or tools that we use to capture what is happening at different levels to make sense of the entire complexity that underlies our genome and to understand the dynamic functions. Now, having appreciated the complexity that we are trying to address, let's first also understand, taking the human transcriptome as a reference, what is the level of knowledge that we currently have? So, as we know, over years of the transcriptome and genomic research that has been happening, a lot of genes have been identified for the human genome. And as of now, there are close to 60,000 genes that have been identified with more than 228,000 transcripts that are coded by these genes. This is as per the latest compilation by the GENcode database. And what we also know that there are different RNA species that we are talking about here. They're protein coding RNA, and we all know that there's a whole set of non-coding RNA which do not end up creating a protein, but then have other regulatory roles performed by them, like the long non-coding RNA, the small non-coding RNA, and other pseudogenes and other kind of genes. Now, if you try to understand, one thing is about discovering genes, while the other thing is to understand what these genes do in the context of the human body and its functions. So if we look at the functional annotation of these genes, again, there is a consortium which has been set up to compile and curate all the research that has been carried out to understand the functions of different genes and their products. And what we know is that close to 20,000 and more of genes and their products, we have some kind of functional annotation known based on what biological process they are involved in, or molecular function they play, or which cellular component they play their role in the cell. So now if you look at the protein coding genes, which is the major section that has been investigated and has been of interest to the research community, you would see that there are most of them, all the green, different shades of green that you see have some kind of functional information known. But then as we go towards the non-coding species of RNA, you see that many of these non-coding species still do not have any function characterized, and which essentially means that we have a lot of unknown non-coding species that we have discovered, but we are still trying to understand its function in the context of different diseases and developmental phases. So this is really one big motivation for researchers to undertake RNA sequencing, because there's a lot of unknown still in that domain in terms of understanding what these transcripts actually do in the body, what role do they play in the different cells and tissues of our body. And the other important factor that we all understand is that transcriptome is dynamic, whereas compared to a DNA, which is almost static, where once you're born with a genetic sequence, you live and you die with that, right? But then transcriptome is very dynamic. And why is it so? Because over the course of development, from a fetus to all the way through your aging process, and also in different tissues and different cell types, although all the cells have the same genetic sequence, we have different cell types, different tissues performing different functions and part of different kind of organ systems, and therefore it's very intuitive that the same genes, that all these cells have different set of genes acting at different points of time to make sure that this kind of wide requirement of different functions are getting delivered. And also we are living in an environment exposed to different kind of stimuli, and these can also impact the way the different genes get transcribed in our body. And definitely something that is most interest is to understand how the genes or the gene expression can be can be altered or regulated or gets regulated in the context of disease manifestations. So these are all the different functional aspects which actually motivate us to understand what and how the transcriptome is changing in these different circumstances. Now, like I said, starting from the 1970s, different technologies have been used to understand, to discover, to identify, and also to measure the expression of different the gene transcripts or isoforms. So starting with the northern blotting and Sanger sequence way back in the 1970s, and then later the hybridization-based techniques which led to PCR, qPCR, and ESTs. Where short sequences are tagged sequences are sequenced to identify the transcript sequences. And then later on, there was a much more economical and a high throughput method that came across, which was the microarrays, where the complementary DNA are identified, based probes are used to identify the transcripts. And then in the early 2000s is when the first RNA sequencing using the next generation sequencing methods were developed, which we will be discussing today primarily, where high resolution, single nucleotide resolution sequencing of RNA was adopted as an approach to understand and to quantitate the transcriptome. And then even beyond that, several disruptive technologies have come into place, which have really, over these 4 decades, a lot of value has been added in the field of transcriptomics, and a lot of biology has been resolved and highlighted using these technologies. The recent focus is obviously to understand these transcriptome at a single cell level, and single cell transcriptomics has been gaining a lot of popularity in terms of being able to tease out the individual cell level functions of the transcriptome. And which is a different topic, a completely different, an entire topic, complete topic in itself, over which I'll not be covering in today's webinar. So as I said, as we speak today, there are different kind of newer technologies that are emerging, which uses different ways of nuances of capturing the expression and the length of the transcriptome in a very accurate manner. So as I said, the long-standing technologies that have been used in the last 2 decades have been microarrays, and recently it has been RNA-seq. Both of them have a lot of advantages and disadvantages, because of which they were used very widely at a certain time point and then later moved on to the other technique. So as I said, as far as microarrays are concerned, it's a hybridization based approach, right? So you have probes put on a plate which will hybridize to the transcripts of interest. And so the pros are that these are low cost assays, low manual labor, and you have very high throughput. You are capturing thousands of genes at a time, and the data analysis also is simpler and more user friendly. Whereas the cons are that in case of microarrays, you are limited by the set of probes that you have put on the chip there. That is, you can only profile the abundance or the presence and the abundance of transcripts that are put on the chip. And that means you cannot do any novel gene discovery or transcript discovery. And therefore, it requires prior knowledge of genes that you want to assay or to profile. And it also does not identify the isoforms or the splice variants. Whereas with RNA-seq, once with the advent of next generation sequencing, we have now single nucleotide resolution, where every nucleotide of the RNA is is read, and therefore it gives you an ability to discover novel genes, novel alleles or isoforms, and it therefore gives you a larger dynamic range to capture. It can even detect single nucleotide variants because you are now operating at the level of single nucleotides. But the cons were, I should say that cost is something that has transformed significantly over the years, that nowadays even RNA sequencing is very much affordable, and it can be very well considered as a low cost option with the use of multiplexing of samples. And yes, because of the amount of data that are generated, it is a complex, much more complex data analysis compared to microarrays, and it's not fully optimized yet. Every year there are improved methods and tools that are being released, which further improves on the accuracy of their detection. And as one would assume with any next generation sequencing technique, because there is a lot of data that is generated and that needs to be analyzed, it requires high computational power and storage requirements as well. Okay, so now having given a brief introduction, now we'll move on to understand what are the major blocks involved in the RNA-seq experimental workflow. So obviously it starts off with isolation of RNA, and you must be already aware of the composition of mammalian RNA samples. So it's important to know that the majority of the RNA species is the ribosomal RNA, and by number of molecules it's the tRNA, and you see that there is about 10 to 15% of tRNAs, and then about 5 to 8% mRNAs, and then the LNCRNAs and other ncRNAs. So why this is important to know is because now when you are beginning to design your experiment, you need to know what species of RNA are you ideally wanting to capture in your experiment, and accordingly you need to know what sort of an enrichment protocol you need to use for your RNA sequencing experiment. So obviously if you are going to sequence this whole RNA, total RNA as it is, it would be easy to imagine that most of the RNA that gets sequenced will also be the dominant species, like the rRNA and the tRNA. And if that is not your interest species, the species of interest, then you are wasting a lot of your money on sequencing something that's not of interest to you. And therefore we have a lot of different enrichment protocols that are being used. So two of the most widely used methods are the poly-A enrichment and the ribosomal rRNA depletion method. So poly-A enrichment is nothing but is a way of enriching using beads, capturing and enriching poly-A tailed mRNAs, because mRNAs are capped and tailed with poly-As. And if you are only interested in understanding or discovering and understanding the expression, measuring the expression of mRNAs, then what you would do is to just go for poly-A enrichment, in which case you have a 76% of your protein coding RNA still being captured with some share of the other RNA species also there. Whereas in case you are interested in all in the other non-coding RNA species also, and because of that, recently the major interest and most of the experiments go for rRNA depletion, where all the ribosomal RNAs are depleted before going ahead with the rest of the protocol, and this enriches all your RNA species of interest. Now, so the basic components of an RNA-seq workflow is what I would like to highlight in this slide. So once you have isolated and enriched your RNA species, the first step is fragmentation of RNA, and this is because usually RNA species are really long, thousands of bases long, and the sequencing platform that we use are most often not able to sequence such long strands of DNA or RNA at once, and therefore it's important to fragment these RNAs into smaller fragments which can be sequenced in your sequencing platform. And then you have the cDNA synthesis, where the RNA is reverse transcribed into the complementary DNA, and this is so that now you are not working anymore with RNA which is not stable, whereas DNA is much more stable and can be altered and modified in a better way. And once you have this double-stranded DNA, the next step in a sequencing platform is to add the adapters. Now, these adapters help the sequencing machine recognize your RNA that needs to be sequenced, and also this also has the primers that will be using the sequencing protocol. Okay, and then the next step is, of course, a PCR amplification, and this is a step where you are going to amplify the entire RNA sample so that you can have enough sample available for sequencing in your sequencing machine. And then you finally have size selection. As I said, your sequencing based on the machine that you are, or the sequencing platform that you are using, they are compatible with sequencing a definite range of fragments, and therefore one has to perform size selection to make sure that you choose the range of sizes that can be now input into your sequencing machine. Now what I described till now is, as you can see, is the set of steps that one would follow for performing a short read sequencing. But as we have different kinds of technologies that are evolving. Now a lot of long read sequencing technologies have evolved, like the PacBio and the MinION. And as you can see, those technologies skip some of these steps. Like if you have the long read DNA technology, obviously fragmenting RNA is not a step that is involved, because now this particular platform is capable of sequencing longer fragments. And similarly, now the latest is direct RNA sequencing, where you don't have to convert the RNA into complementary DNA before sequencing, where the RNA can be directly sequenced. So as I said, although these are the basic steps that are involved in a typical RNA-seq workflow, depending on the experimental the sequencing platform that is available to you or that you want to use, there would be changes, corresponding changes in the workflow. So as I said, the one that I described is mainly for the Illumina based sequencing platform, and but we also have Pacific Biosciences and nanopore based technologies which are offering long read sequencing. So in Illumina, the typical sequencing principle that is used is the sequencing by synthesis principle, where fluorescence tagged bases are flown into the flow cell, where every nucleotide is synthesized as you go, cycle by cycle, and based on which nucleotide is attached to the or hybridized to the template sequence, a corresponding light is flashed, and which is used to call your bases. Now, in case of Pacific Biosciences, they use a different technology altogether, where you generate circular templates by the ligation of specific adapters, and then you are capable of now reading longer sequences. And then again, there is a light-based flashing. That's a signal. That signal is generated based on fluorescence tagged bases. And then in case of nanopore, it's another nanopore technology is where the protein nanopore is embedded into the membrane, and then the DNA is processed through these nanopores, allowing for again long lengths of sequences to be sequenced, fragments to be sequenced, and thereby giving you longer read lengths. So, before I proceed further, I think it's important that we discuss some of the important terminologies that we repeatedly use in the context of RNA sequencing. One is, of course, the read, which is nothing but a sequence of nucleotides that is sequenced from the fragments that we input into the sequencing machine. And so we have two different kinds of sequencing, the single-end sequencing and the paired-end sequencing. Single-end sequencing is where a fragment that is input into your sequencing machine is read only from one end to the other, like from 5' to the 3'. But paired-end sequencing is where the fragment is read from both ends, and I will tell you how that helps. So there are several advantages of paired-end sequencing, although it is a little more expensive than the single-end sequencing, which is that it gets easier to now study the splicing isoforms that we discussed earlier, and also it provides improved accuracy in mapping to repeat sequences as well as exon-exon junctions, because now we have two paired sequences that you are always trying to align back to your genome, and knowing that these two pairs are originating from the same fragment separated by a certain distance improves your accuracy of alignment and calling of transcripts. And also in case of de novo transcriptome sequencing, this is when you do not know anything about the reference genome, and you are trying to assemble the entire transcriptome of a new species probably. And even in such cases, you need to have paired-end sequencing as a technique used, because that provides improved accuracy when you are trying to generate your contigs, to assemble your contigs from the individual reads. Now, the other concept, I kept talking about short read and long read. So the whole concept of read length is very important in RNA sequencing, which is nothing but the number of nucleotides that are sequenced per read. So you have 75 nucleotide, 150 nucleotide, 500 nucleotide, and now, as I said, the latest technologies have thousands of bases that can be accurately sequenced. And the pros of having longer read lengths, as you would guess, is that again, because the isoforms, the transcripts are spanning multiple exon junctions, having longer read sequencing makes it easier or more accurate to call such isoforms in complete length. And also in case of de novo transcriptome sequencing, it's very important, or it's very beneficial when you have longer read sequences. Now, another important terminology that is often discussed in this context is the sequencing depth or the coverage, and that is nothing but the number of times each nucleotide on the reference genome has been read on an average. So, just to explain it a little more, let's say this is the red is the reference, the red block is the reference genome, and the blue blocks are your reads. So now let's come to the first one. So for this position on the reference genome, you can see that there is only one read mapped to that position, right? So that position has been mapped once. Okay, so the coverage for that position is 1x. Whereas if you come to the second position here, there are two reads mapped to that position, so the coverage is 2x. And if you come to the third one block here, there are four reads mapping that position, so you say the 4x coverage for that particular position. Whereas there are several positions on the genome where you have 0x, that is no read has mapped to that position. So this 1x, 2x, or 4x, that tells you how many times a particular position on the genome has been covered. And the sequencing depth of a particular run is nothing but on an average how many times every nucleotide on the reference genome has been sequenced. And why is this important? So that coverage can be of course calculated as the total number of bases that you sequence and divided by the size of your reference transcriptome. That will tell you on an average how many times each position is covered. And why is this important is that adequate sequencing depth is very important when you are trying to discover and also quantitate low expressing transcripts, because as you can imagine, the low expressing transcript numbers would be of very low abundance in the total RNA sample that you are trying to sequence. And therefore, if you don't have enough reads covering a low expressing transcript, it would be very difficult to capture them in the first place. So as I said, increasing the number of reads that you want to sequence gives you an advantage of being able to identify or detect novel or newer transcripts or low expressing transcripts. But then there is always another factor that needs to be considered, which is the number of replicates that one would use in an RNA sequencing experiment. And so what has been shown is that if you want to understand whether, if you, let's say you have a certain amount of money with you and you want to know where you want to do the trade off, do you want to increase the sequencing depth, that is, do you want to generate more reads from your sample, or you want to sequence more samples, use that money to actually do more replicates of that particular condition. So there are some interesting insights that were generated from this study from Liu and its paper in 2014. So this really depends on which kind of transcripts that you are wanting to capture. So for example, in the first block here, which is for high expressing genes, you see that beyond 10 million reads, there is not much improvement in the total number of differentially expressed genes that you would identify. But whenever you increase your number of replicates, that significantly improves the total number of differentially expressed genes that you would identify from your experiment. So in this particular case, it is a no-brainer that increasing replicates actually helps you find more meaningful differentially expressed genes than actually increasing your sequencing depth. But when it comes to low expressing genes, like in the lower plot that you are seeing here, you see that as there is actually an advantage in increasing both sequencing depth as well as the number of replicates. So this gives you a perspective of these are important consideration while deciding what kind of an experiment you want to plan. when you are doing your RNA-seq experiments. Now I'm going to move to the data analysis part of it. Just to give a snapshot of what is involved in an RNA-seq data analysis, this is what it is. You have the raw RNA-seq reads that you obtain after your sequencing run, which definitely needs to undergo quality control. As I said, quality control is something that needs to be done at every step of this entire workflow, not just for the data analysis as well as for the experimental workflow. Even in the experimental workflow, at every stage you do different kind of assays to judge or to evaluate the integrity of the RNA, the library, the size, the clustering, and different aspects of it. So that is something that is definitely not to be missed. So as I said, even in data analysis, it's very important to identify all the garbage reads, to make it into the most high quality reads before you go ahead and try to process any of them. There are several set of tools that are available for all of these, which we won't have time to go into all of those. But this is just a snapshot, as I said. And once you have the high quality reads, the processed reads, then you go ahead with what is known as reference mapping, which is nothing but you have your reads now, and if you, whichever species your reads have been generated for, now you go fetch the reference genome for that species and you map it back to the read. And because that, only once you do that, you can now start to assemble your transcripts or your genes that are present in these, or that are captured by these reads. And finally, you go and once you quantify the abundance of these transcripts, we go ahead and do the differential expression analysis or the other analysis goals that you have from your RNA sequencing experiment. So the raw data, if you want to just have a look of the raw data that is generated from the sequencing platform, this is how it would look. Each color represents one read. So there are 4 lines to one read. Okay, so each read is represented by 4 lines in that FASTQ file that is generated out of the sequencing machine. The first line starts with an @ symbol, which is the header that gives the FASTQ header, which gives the unique sequence identifier for that read. And then you have the second line, which gives the basis of the read sequence. And then you have the third line, which is just a plus, and the fourth line, which is all those characters that gives you the quality scores of each base call of the sequence. So like I said, like at least in case of Illumina sequence, we briefly discussed the sequencing by synthesis. So as I said, at every cycle of the nucleotide flow, the light signals, the intensity is measured based on which nucleotides are added. And at any point of time, if the intensity is low, or there is low diversity in that entire chip, you can have lower quality scores, which tells you that there was less confidence in calling that particular base. And that is very important in terms of calling the transcripts also accurately downstream in our analysis. So these are the major components of the data that comes out of the sequencing machine. And then, as I said, the second step is, of course, to do the quality control and pre-processing. So there are several aspects that you want to analyze for this particular action, which is the per base quality score. So that looks something like this on the right. So let's say you have a 140 base pair long read, like shown here, 150 base pair long read, and what you're seeing is the quality scores for each of those bases averaged across multiple reads that have been sequenced in that run. So as you can see, towards the end of the sequencing, because the efficiency of the enzymes are also deteriorating towards the end of your sequencing run, there is a high chance that the quality scores of the bases that are called tend to drop. And it's important to trim those bad quality basis of your read so that your downstream analysis becomes very robust. So there are tools that allow you to understand, to analyze these different parameters that I am talking about, like the mean quality scores. Each read can have a mean quality score, and that is again a parameter that can be used to judge if a particular read should be taken for downstream analysis or not. You might decide to have a cutoff saying if the mean quality score is less than a particular threshold that you set, I am going to throw away those reads, those garbage reads. Then another important parameter is the sequence GC content. Again, GC content is something that helps you understand if there is some kind of contamination in your sample, or a certain species, RNA species overrepresented, if your RNA depletion has not worked, or different kind of aspects like that. So having an abnormal GC peak can again give you hints about how your prior protocols have gone well or not. And then the adapter content, as we remember in the experimental protocol, we added adapters, and it is important to remove adapters before you go into your downstream analysis, because those sequences are extraneous to your genome and those are not going to map back to your genome. So it is important to remove those, trim those adapters. And another is again contamination. There is another tool called FastQ screen, which actually lets you assess contamination by taking a random subsample of your sequencing reads and mapping it to different genomes, and seeing if the reads are mapping to the genome of your interest indeed. Otherwise, it can, if it goes and maps to some other species, then that again gives you an indication that there was some kind of contamination in your process. And again, another metric that is used to judge the quality is percentage reads that map to the reference genome. So if you have sequenced a human genome, and if you don't have at least 70 to 80% mapping to the human genome, then definitely there's something wrong with it, and that needs investigation. Then another aspect that is looked at is genomic origin of the reads, which is if the reads indeed originated from the exonic part, because we have sequenced the transcriptome, and therefore you look forward to having a lot of exonic regions in your reads and not the intronic regions, and that looking at a spread of the distribution of the exonic versus other regions of the genome gives you again an indication of whether your protocols have worked fine or not. And similarly, percentage PCR duplicate reads, that's also an important metric to judge if you have not artificially created any amplification or duplication of your reads, which can again affect your transcript abundance estimations. Now we go over to the next step, which is alignment. As I said, alignment is a very important step where you have to now, after you have obtained your high quality reads, the processed reads, you are going to now map back these reads to the genome to understand, so that you can now call your or identify the coordinates of your gene and then estimates its abundance. Now, there are two major methods of alignment that are being used now. One, and the most important one, and most widely used one, is a splice-aware reference-based alignment. So, as we already saw, since we are going to map back RNA and not DNA reads, right? So, what is special about the RNA reads is that you will have reads that are originating only the exons. So, basically, you will have reads that are mapping to exon junctions. And when you map that read back to the genome, it is not going to map to the intron in between. The genome will still have the intron, but it is not going to map to the intron. It will only map to the two sides of the exon as shown here. So your alignment tool needs to be aware of these exon junctions and allow for the read to match in a split manner this way. And this is not a mismatch. This is how it would match because it is an exon junction. So this kind of alignment is known as splice aware reference based alignment, where the reads can be mapped across splice junctions ignoring the introns, and this is very CPU and time intensive and memory intensive. And some of the tools that do this kind of alignment are Top Hat, HiSat, and STAR. Now, the other kind of alignment that has recently emerged is the pseudo alignment, which stems from this idea that you don't really need to align it back to the genome. If I have a reference transcriptome, why don't I just align my reads back to the transcriptome? Then there is no exon junction to be. This doesn't need to be splice aware. It can just directly map to the reference transcriptome, and you are just matching for compatibility with the transcripts, and then you can immediately very easily compute the abundance also thereafter. And this sort of an approach is, as you would assume, it's extremely fast and it's very lightweight in terms of the memory footprint that it leaves. So the tools that does this kind of alignment are Salmon and Callisto. So these are tools that you can run very easily on your personal computers, in a matter of minutes and hours, which otherwise takes hours on a cluster. Now, after you have aligned, the next important step is the transcriptome assembly, where you now start calling the transcripts, which is nothing but defining where it starts, the start and end of each exon of the transcript that is represented by your aligned reads. So again, there are two different ways you can do the transcriptome assembly. One is a reference-based transcriptome assembly, where you use a reference gene annotation. So in species like human, where the transcriptome is quite well characterized already through the multiple research initiatives that have happened across the world. It is way of lot of interest to use the reference genome annotation. It's going to improve the transcriptome assembly. So therefore it assigns reads to the genes, and then it gets the read counts per gene or transcript definition, which you are providing upfront. But then now there are also species where we don't have enough information on the reference genome or the transcriptome, in which case one has to go for de novo transcriptome assembly, where you don't have any reference genome or transcriptome. You are just trying to overlap the reads and assemble reads into gene contigs, and then assign the reads to these gene contigs and annotate the contigs, and then get the read counts for each of these gene contigs. Again, the transcriptome assembly can be performed by different set of tools like cufflinks, HT-seq. There are several others that can help you do all these steps. And then the next important part. So once you have counted the number of reads for each of these genes or transcripts, next, the most important step is to perform normalization. Now, why is normalization important? Normalization is important because when you are having different genes in your transcriptome of varying sizes, and it is important to normalize for their lengths. For example, a longer gene is likely to have more reads mapping to it compared to a shorter gene, and therefore if you don't normalize for the length of that gene, you are likely to make a wrong conclusion that the longer gene always have more expression or more read counts than the shorter gene. And therefore different normalization methods have been developed, and read length is the gene length is just one of those aspects. Another important aspect is the library size, or the number of mapped reads per sample. Let's say we have two samples, and in sample 1 you have 10 million reads, totally 10 million reads mapped to that sample, whereas in sample 2 there are 20 million reads. It may be because the sample 1 had more low quality reads, which we removed in your pre-processing step, and therefore it was left only with 10 million good quality reads, whereas the sample 2 happened to have 20 million reads. Now when you map the reads to the same genes, gene A and gene B, it is you are likely to see that gene A and gene B of the second sample has more reads compared to gene A and gene B of the first sample. But then it would not be right to conclude that the sample 2 has higher expression of gene A and gene B compared to sample 1, because the sample 2 had more reads mapping to it to start off. And therefore here in this particular case, it is important to normalize also for the total number of reads that have mapped for each of those samples. So as I said, these two are one of the most important parameters for which one has to normalize the read counts that you obtain for each of the genes. And there are several. After normalization, you have different metrics like the RPKM or FPKM, which are used to indicate to define the abundance of a sample. Like for example, RPKM is reads per kilobase of exon per million reads mapped. Okay, what that does is for each particular gene, you would take the total number of reads of that particular region and divide it by the total mapped reads for that sample into region length by 1000. So this basically gives you a number that has been normalized for the total maps read reads map, as well as for the gene length that is involved. Similarly, there are other normalization methods that are used, like the FPKM. Again, that's another measurement for the transcript abundance. Again, all generating from the read counts for the genes. Then you have then you have TPM, that is another measurement that is often used, and TMM, which is trimmed mean of M values. Again, all these are different methods that use different normalization approaches for accurately estimating the abundance of the gene. Now, so what finally you get after this transcriptome assembly and quantification is basically a matrix or a table where you have all your genes or all your transcripts in your rows and all your samples as columns, right? So if you have 20,000 genes, you have 20,000 genes, and then how many ever samples that you sequenced. So one of the first things that I do, or that one should do after you get this kind of a table, is that you need to do what you call a principal component analysis to understand how your samples cluster based on their whole gene expression profile. So PCA, or principal component analysis, is a dimensionality reduction analysis tool which helps you plot or visualize the samples based on their overall gene expression profile. So what that helps you do is, so it is to generate plots like this, where each dot will be a sample. Okay. And then, so in this particular assay, you can see there are thousands of samples that have been assayed. And what this lets you see is how the samples cluster based on their gene expression profile. So what the most important utility of doing this kind of an exercise is, it helps you visualize or get an overview of whether the different groups of samples that you have sequenced as part of your experiment are clustering together, or are there some samples that are outliers which don't cluster with any of the groups, and they are outliers and which need to be excluded for any of your downstream analysis. And another important factor is to see if your data has any batch effects. That is, let's say if you had a very wrong kind of experimental design where all the controls were sequenced on one day and all your cases samples were sequenced on another day, then you are likely to have a separation between the two clusters just by virtue of the fact that those were processed on different days, right? So those kind of batch effects, the technical artificially induced clusters, can be identified by using this kind of an analysis. And it's very important to do this kind of an analysis before you proceed into any of your downstream analysis, because that lets you correct for these kind of errors, and also to remove the outliers which can otherwise have an impact on the results of your analysis. And then it's important to also know what are the different analysis goals. Depending on that different tools, you might choose a different set of tools. So obviously one of the most popular thing, the application for which RNA-seq is used, is for understanding differential expression. You have two different conditions or two different disease stages or two exposures that you want to compare, and you do a typical differential expression analysis for doing that. Another could be you want to identify some new genes that are implicating its function in a particular situation, or to understand the function of some genes. You have a gene knocked out and a gene, a normal sample, and you want to understand what is happening when a particular gene is knocked out. So those are explorations for understanding what happens if a particular gene is not there, right? So there are different asks to different experiments that you run. Another could be alternative isoform expression. Like I said, the same gene can give rise to multiple isoforms based on the different exons that are stitched together. And you might be interested in knowing which is the major isoform that gets expressed. Are there different isoforms that gets expressed equally or under different conditions? Maybe in a healthy condition it is one isoform, and in another condition it's another isoform. So you want to know these kind of alternative expression. Then allele specific expression, that is when you have, let's say, a mutation. Because of the presence of a mutation in the gene, maybe a certain isoform is expressed more than the other. So those kind of differences you want to see. Or you want to look at gene fusion. So as typical in different cancer cases, you have different genes fused together, and you are interested in looking at which are those gene fusions, and there would be a different pipeline that you would adopt for identifying those kind of things. Then mutation discovery and RNA editing. There are post transcriptional RNA editing, which is also another regulatory process that is involved, and one might be interested in exploring those. So again, there are a different set of tools that you would use based on what your key goals are for from your RNA-seq data. So going to differential expression analysis, as I said, the objective is to identify the genes which show a statistically significant difference in their counts or the expression between two or more conditions. And as I said, it's not just important for it to have a significant difference, it should be statistically significant, where the p-value is less than 0.05, which means, there is only a 5% chance that that particular difference happens by random chance. And for differential expression analysis, the conventional student's t-test is not employed in case of RNA-seq analysis, because the gene expression count data is often not normally distributed, which is a basic assumption for any t-test based analysis. And therefore, the method that is widely used for differential expression analysis is negative binomial based methods, and these are the methods that are used in EDGAR, DESeq, CUFDIF. So, why I'm talking about this is when you adopt a particular tool or a pipeline, it is important to understand which are the different algorithms and the statistical tests that are used in these tools, which helps you make a good call over which tool has to be used for your analysis goal. And as I said, over the years, several tools have been released for the purpose of doing this entire sequence of events that I just described as part of the data analysis of RNA-seq. As you can see, there is a different set of read aligners that you can use, different set of expression modelers or the transcription assembly modules that can be used, different kind of differential expression tools that can be used. So when you are doing a particular analysis, you might be choosing a one particular combination of these three kinds of tools. And there was a recent paper which actually looked at evaluation of the precision and recall of these different pipelines. So taking each one from, like, for all combinations of these three pipelines, and what they found is that there is always a trade-off between precision and recall when you use a particular kind of tool. And therefore, it is very important to understand what is your requirement and which is the specific set combination of tools that you want to use for addressing the requirements or the questions of your problem. And another important factor is multiple testing correction, because remember, we are actually testing 20,000 genes for differential expression analysis, and therefore when you are doing multiple statistical tests, it needs an additional correction, and that is usually performed using false discovery rate. Bonferroni is another method which is a more stringent approach, whereas false discovery rate is the method that is used to additionally ensure that the results that you infer from these kind of large high throughput analysis is statistically significant and functionally relevant, and that less of false discoveries are present in your analysis. And once you another important aspect of the differential expression analysis, of course, to visualize the expression of the differentially expressed genes, we use heat maps to do that. We also represent the differentially expressed genes using what we call a volcano plot, where the fold change, that is the number of the number of times the change in the gene expression between two conditions, the twofold or fourfold or half, right? So the fold change is plotted against your p-value, and whatever comes on top and towards the extreme, the blue colored ones, that's the threshold that you set to identify your differentially expressed genes from your analysis. And similarly, once you identify your differentially expressed genes, then you go and you are interested in understanding what's the functional significance of these genes. Fine, I identify 200 genes. Now, what are these 200 genes representing of? Are they all of a particular biological process? Do they all originate from a particular cellular component? And what function do they perform in the body? Is there a particular pathway that these genes represent? So these kind of analysis further helps you tie your results to the actual biological answer that you are looking for. Another kind of downstream analysis that you perform is what you call the co-expression network analysis, which is again trying to correlate similar expressing genes with similar expression patterns. And the whole hypothesis is that the associating genes often originate from same pathways, and therefore that is used as a way to annotate, to assign new functions to unknown genes. So this co-expression network analysis again is a very important, very powerful tool that lets you infer a lot of interesting insights from your analysis, from your samples. And after having given an overview of the data analysis, now I would like to share a few useful RNA-seq resources that you might be interested in looking at. One obviously is the Gene Expression Omnibus. This is a repository of all the transcriptome data sets that have been submitted by researchers, all around the world. There are, as you can see, there are more than 4,300 different data sets, or more than that possible, that are being submitted. And these are publicly available data sets which can be combined and analyzed in different ways based on the unique question that you want to ask. And different interesting insights can be inferred from doing these kind of analysis of large scale analysis of public data sets. So GEO is one place where you would look for such publicly shared transcriptome data sets. And another important initiative that happened was the genotype tissue expression portal. So as I said, we have several hundred different types, cell types, and different tissue types in our body. And understandably, different genes are expressed to different levels in each of these genes. Apart from the housekeeping genes, there are several genes that have spatiotemporal expression patterns. And this is one database which will let you investigate genes from that point of view, in terms of understanding which are the tissues and cell types where a particular gene is expressed more or expressed less. And that can be very useful for undertaking your and expanding your research insights. Another very important study of the last decade has been the Encode Project, where all the different elements that I discussed in the first slide of my presentation, which play a very important role towards regulating the function of the different genes in different cell types and conditions, have been profiled using multiple tools. And this is one place where you can appreciate the power of integrating different levels of omics data sets for generating some advanced biological inferences, because the power of combining, let's say, a methylation data set with a transcriptome data set along with a chromatin accessibility data set, it adds different levels of evidences which can really help you cement all those hypotheses in a very confident manner. So if you are interested in exploring integrative omics as an approach to understanding, solving different complex biological problems, this is one place where you can derive a lot of ideas. I think with that, I will conclude today's session, and I'm really happy to.
Thank you so much, Dr. Ambily, for such an insightful talk. Thank you all for participating in this webinar. If you are watching this video on YouTube, thank you so much for watching. Please like our video and subscribe our YouTube channel. If you have any questions related to the subject, you are always welcome to contact us through mail, or you can connect with us through different social media handles. This was your host Diksha Bhartiya on behalf of the team GENoMiX, now signing out, and have a nice day.